We've seen that central dogma of molecular biology is when DNA could be transcribed into an RNA molecule, and this RNA molecule travels outside the nucleus, goes to the ribosome, and gets translated to create a protein. However, this is not. We have other kinds of stuff surrounding the central dogma. Some researchers says that the central dogma is no longer. How can I say? It's no longer useful for predicting this, because we have so many things around this central dogma that it's not like we shouldn't understand that it's all that's happening inside the cell. There's a lot of things going around it. So for example, chromatin can also have, or the DNA could also have small modifications in their histones. If you remember, histones is a protein complex that gives chromatin structure. So the DNA winds around these histones and then goes to the next and then the next, and this helps to give structure to this DNA. So we can have modifications in these histones. As you can see, we can have a phosphate group, or we can have another carbon or another thing, or we can also have modifications strictly in the DNA, especially in the cytosines, where it can gain a functional group. These epigenetic markers, we call them epigenetic markers, that can be edited, have regulatory roles. Recently, it has been found that these epigenetic modifications can be inheritable also, and it can be heritable besides the regulation roles in gene expression. When I say gene expression, I mean the rate of transcription of a gene. So they're very, very important because of these regulatory actions that it can have, and because it can be inheritable. So it can be also, they can be modified and stuff. Well, then we can, transcription by now. However, we can have retrotranscription also. It's not that common. However, retrotranscription is a process to construct a DNA molecule from an RNA molecule that is integrated. Well, in some cases, for example, in some viruses, for example, in the immunodeficiency virus, that is, or HIV. HIV has RNA molecules inside, and it can retrotranscribe this RNA and create a DNA molecule which could be integrated into the human DNA. That's what it's called, a virus. And after being integrated, it could be transcribed again, and this is how HIV gets reproduced. However, retrotranscription is also present in eukaryotic cells, as the human cell in retrotransposomes. These are movable regions in DNA. However, we're not going to talk a lot about that. However, you should know that retrotranscription is also happening inside human cells and some other kind of animals and prokaryotes. However, it's not that common as transcription is. We also have, we have already talked about mRNA. However, there are other kinds of RNA different RNAs that are involved in messenger RNA silencing or other kinds of functions. For example, the ribosome has these pieces called transfer RNAs, which we're going to go a little deeper in a while. And we also have RNA processing. RNA molecules can be modified before reaching to the ribosome. They can be cutted and pasted. They can be added some nucleotides and some other process before going to before getting out of the nucleus and going to the ribosome. We also can have protein modification because in this way, when a protein is modified, it can achieve some other functions that the first protein didn't have. So this is important for you to know also. So about epigenetics, there are two main large projects on epigenetics. The first one is called the National Institute of Health, or NIH, Roadmap Epigenomics, that we have their logo here. And it is made in the United States, and the Roadmap Epigenomics project expects to deliver a collaboration of normal epigenomes that will provide a framework or reference for comparison and integration with epigenomic differences in diseases. All of this information is freely accessible online. So this project can give us this framework of a normal and a healthy cell, and that this framework could give us a reference for future analyzing epigenetic mutations could be called, or the epigenetic modifications that could give rise to disease. So that's very important. They analyze different kinds of epigenetic marks, like DNA methylation. Methylation is a process where a carbon and free hydrogens is added to DNA, especially in cytosine base. They also analyze histone modifications and RNA expression, and how these modifications are involved in expression. There's also the Human Epigenome Project that is in Europe, mainly in England. This project aims to identify, catalog, and interpret genome-wide DNA methylation patterns of all human genes in all major tissues. They're analyzing different kinds of tissues and how they are marked in their epigenomes. They are mainly analyzing DNA methylation only. As you can see, well, I have here this cytosine, normal cytosine, and 5-methyl cytosine that in the number 5 carbon it's been added a methyl group. So it's been observed. Well, before that, this Human Epigenome Project is also it's a public and a private consortium in Europe. DNA methylation is very important because it can be modified by different influences, which means that environment can change it, and this influence the gene expression. Differentially methylated regions give rise to distinct patterns that are specific for tissue and type and disease state. So these epigenetic markers, for example, in here, if these cytosines are marked with a methyl group, it can lower the gene expression rate and thus give rise to some disease in some kinds of cells. So it's been very important. These epigenetics have been a huge boom in these recent years due to the recent techniques that have allowed us to measure this, where we have these marks and where we don't have them.